Okay, good afternoon, everybody. Welcome back to the Innovative Discoveries series. To finish out the academic year, we have really an exciting talk today. But remember, for CME credit, you must first sign in and must include your email and credentials. So, great Independence Day weekend next week. We'll be back on July the 6th for a Thursday Tech Talk with Paul Neitert talking about knowledge translation. Please see the website for the full schedule. It's really a great thrill today to introduce my very good friend, Cathy Wu from the University of Delaware. She is, as you can see, the Edward G. Jefferson Chair and Director of the Center for Bioinformatics and Computational Biology at the University of Delaware. She did her undergraduate work in Taiwan and got her master's and PhD at Purdue, and then did a postdoc in molecular biology at Michigan State, followed by a master's in computer science, I should say, at the University of Texas. And so she's really well-grounded in both biology, molecular biology, and computer science. She has been working in computer science and computational biology at the very highest level for some 25 years. She has some 250 peer-reviewed publications, multiple NIH grants at any one time. She's also a terrific teacher and lecturer. I've heard her lecture before, and it's always remarkable to hear. And she has a PhD program in bioinformatics that she also has at the University of Delaware. It's really a very great thrill today to introduce our very good friend, Dr. Cathy Wu.
Well, thank you, Bill. That's a wonderful introduction. Very nice of you. So I'm really thrilled to be here to talk with you about very exciting research that we have conducted at Center for Bioinformatics and Computational Biology, and also at the Protein Information Resource. So, certainly, this is a very exciting time to do research in the area of big data and bioinformatics, and see how that could relate to systems biology and precision medicine. How that could potentially, the kind of knowledge that we will gain from the big data could translate into some very important applications, scientific applications in basic biology understanding and in medicine. So just outline, the first part is more introduction on big data and bioinformatics, how sequencing, next generation sequencing really has transformed how we are conducting biology, becoming a big data scientific discipline. And then I will focus the discussion about the kind of bioinformatics, medical informatics infrastructure that we are building on, focusing on the literature mining project. Basically, how do we extract information from the scientific literature and then convert that into some kind of knowledge framework where we can connect genes, disease, and drug information. And the last part, I would like to show you a couple scientific applications, in this case focus on the knowledge networks that we can build using information from post-translation modifications, the kinases, the substrate relationship, the gene regulatory network, the protein-protein interaction network, and how that kind of knowledge network that we build by gleaning information from scientific literature and integrate data from many disparate resources could then be used as a knowledge base to try to interpret a lot of the omics data being generated, whether we are talking about genomic variant data, whether we are talking about the expression, the RNA, microRNA expression data, the proteomic data, the phosphoproteomic data. How do we interpret those kind of omics data using the knowledge network? And specifically because we talk about kinases, substrates, the phosphorylation, and so forth, so I will particularly highlight our application areas, the collaborations that we have conducted in the area of kinase inhibitor drug analytics. So that's, and how we work with physicians and with our collaborators in really turn the kind of knowledge base that we are building to something that could be used for basically inferencing, knowledge discovery. So just as an introduction, so certainly we are all very aware of how biology has been transformed by the sequencing technology, how the cost per genome continues to go down dramatically, outpacing the Moore's law. And at UD, we have state-of-the-art sequencing facilities. And very excitingly, actually, this fall, we're going to have a new toy, a second-generation PacBio called SQL, that will allow us to do really very long, sequencing. And the capacity is going to continue to outpace what we were able to do before. But it really points to this change in terms of how biologists are doing biology these days, in that the sequencing is no longer a specialty tool limited to only those large sequencing centers. Labs, large and small, can use the kind of sequencers that we have to generate huge number of volume of data related to whole genomes, exome sequencing, regarding transcriptomic profiling to look at gene expressions, proteomics, mass spectrometry, certainly also the technology continue to take off and advance. And so that really is leading to this sequencing-driven biology era. And so biologists, researchers, are getting very, very good in generating a lot of data. But the key here is that how can we convert those data into some useful knowledge for discovery? And if we can translate that into patient care, more precision type of prediction and therapy regarding the patient disease, that would be really great in terms of a goal as to how this can really take us towards patient care. So we need to know how we can really utilize this kind of data that we are so good at generating and really convert them to knowledge. And so there are many different kind of analytical tools. Certainly, from this RNA-seq data, Affymetrix microarray data regarding the gene expression, we need to go through, do different kind of statistical analysis, but also we need to look into the functional interpretation of how those genes, those genomic variants, how those different forms of the proteins, they actually connect in the biological context, in pathways, in networks, and so forth. Because those genes, proteins, they don't act alone in the biological system. So we have to look at how they actually interplay, interconnect in a biological system. And so that's how the individual data items regarding the genes could then be translated into something that will allow us to have better understanding of the underlying biology, potentially the molecular mechanisms underpinning disease processes and so forth. So we are facing with this data analysis bottleneck, and we really need to have more trained bioinformatics researchers to allow us to really best utilize all those data being generated and could be analyzed and reanalyzed, integrated, and ask new questions, generate new hypotheses. So if we look at the biology, driving force in 20th century biology has been reductionism. So we all hear about the Human Genome Project, how we wanted to look at individual parts. We wanted to drill down to individual cells, individual molecules, individual genes, sequences, and so forth. And so it has been driven by reductionists so that we really get very well the part list. But now we have all the part lists. We wanted to see how the part lists actually get connected through integration. So we wanted to connect genes into gene networks, metabolites into metabolic networks, cells into organisms, individuals into ecosystems, into community. And so that really is a concept of systems biology, the birth of omics, from individual genes, parts, to genomes, individual proteins to proteomes, individual protein-protein interactions to interactomes, individual metabolites to metabolomes, and how that could be fit into this hierarchical structure from individual to population. And that really is, again, the basic concept of systems biology, how those different players in a biological life system could be brought together and look at how they interact at different levels of complexity and organization. So medicine, the same way, certainly we are looking at systems medicine approach where we don't just look at symptoms on individual organs or individual tissue and so forth. More and more, we would like to look at systems from the more holistic view, focus on the interactions and dynamics, look into the interactions between genes and environments. And so we will be looking at the genomes, the environments, the gene-environment interaction, and how that gets manifested in these complex biological networks, and how that could then be utilized towards more precision medicine, more personalized medicine. So Precision Medicine Initiative was launched a couple years ago, and so the timing is really right. People have been, the concept is not new. However, to really translate those concepts into an initiative and put the community behind this initiative to work together to generate meaningful data from this precision initiative, it's really great timing that we are doing it now. So we have much greater understanding about the human genome, and not only human genome, but also many other model organisms. And people are engaged in healthcare and research. We have all sorts of sensors, Fitbits, you monitor your activity and your lifestyle and so forth. And so those data are there, sensor data are there that will allow us to look into the interplay of the genes and the lifestyle and the environment that you live in, the air quality, those kind of information. And certainly the data is much more shareable and interoperable because, again, the push in making sure the data is presented in such a way that in such common format that they could be interoperating with each other. And certainly research technology, all this sequencing, all this omics technology continue to advance. And so that really is a perfect timing for us to then looking into patient care, medical care, treatments, no longer just as average population, but more genomic, looking at individual genomic environmental lifestyle differences that allow us to more precisely prevent and treat disease. And certainly this has been very much the kind of practice, especially in oncology, where you would be looking at treatment tailored to specific genes and so forth. ER positive, negative, get different kind of treatment and so forth. But this is certainly expanded in a much broader sense from oncology to other field of clinical care. And Cancer Moonshot, we also heard about it. Certainly, Biden initiative, Cancer Moonshot here. And again, data plays a very much central role in this Moonshot initiative as well. So there was this blue panel recommended, made 10 major recommendations, and one of them is this national cancer data ecosystem. And so the idea is that, again, if our data, our tools can interoperate, we can do something much greater than any individual of us would be able to do. And so at this biology as a big data science era, the era of scientific big data, we do have a lot of opportunities to build the kind of infrastructure that will allow us to translate much more this data to knowledge. And so at Protein Information Resource, this is a resource that I direct. We have many investigators and our professional staff. We developed this international resource, PIR, that provide integrated bioinformatics resource for genomic, proteomic, and systems biology research. And our group have worked on aspects that really relates to data integration, relates to omics analysis, visualization. We also use biomedical text mining, which is a topic that I will be covering quite a bit today, and using ontology to foster the interoperability of underlying data. And we are part of national, international collaborative networks like UniProt and Proteomics Consortium that allow us to really leverage our consortium's infrastructure and our scientific expertise to develop great infrastructure and resources. And we do have a huge scientific community who are accessing the resources that we developed from PR and UniProt with more than 500 unique sites worldwide on a monthly basis, with more than 5 million hits a month. So UniProt, I don't know how many of you have used UniProt. Basically, it is a central hub for protein sequence and function. And so we have, again, this international consortium that consists of us in the United States and our partner institutions in UK, Cambridge, UK, and Geneva, Switzerland. And we present our resource as a central hub where people can connect protein information. And our curators, we have more than 100 curators from the 3 sites collectively. We curate from scientific literature the most salient information about every protein, for all the human proteins that have been known, characterized. We collect all those information, and the information are represented with many different sections there, including the function and information regarding function and also features in terms of if they are protein structure, if we know about binding sites, if they are clinically related, genomic variants, nature variant, and the related pathology information. All those kind of information are captured in this central database. And you can look at how the different type of sequence features, including the binding sites, the catalytic sites, active sites, different kind of important motif and information, how that correlate with clinical variant. And so we have provide that kind of map all these kind of important protein functional sites, structure sites, modification sites to genomic variants. And we can look at, this is just as an example, there's a particular gene, GLA gene associated with Fabry disease, and we can put them on Genome Browser, which is a kind of browser used by a lot of genomic scientists who look at the kind of clinical genomics data. They wanted to look at individual genomic variants and what potential disruption the variant may be causing that leads to disease state. And so we can look at active site information, disulfide bond information, glycosylation site, just as an example. And so, for example, this glycosylation site, the variant disrupt the site for lysosome targeting, therefore leading to pathogenic disease phenotype. And so that's the kind of information that we provide through the UniProt Consortium, just as an example. At the Center for Bioinformatics and Computational Biology, this is a center that I established now almost like 9 years ago, since I joined University of Delaware. And certainly we have wonderful affiliate faculty from across college and our partner institutions. As Bill mentioned, we have our master's and PhD program. And at the end, I'm going to show you we also launched an online, fully online applied bioinformatics certificate program with some information there if you are interested. And we also provide bioinformatics core where really we focus on the capability of analyzing the genomics data, next generation sequencing data, and use that capability to work with our investigators, again, to provide functional interpretation of valuable omics data they generate. So we have quite extensive collaborations with different groups, and certainly we work with many computer scientists. So this is the type of work that we do. The literature to knowledge, the one that I'm going to highlight today, this is in collaboration with our colleague in the computer science department, Vijay Shankar, who is expert in computational linguistics, natural language processing. And so we have worked for more than 10 years now and developed the kind of workflow, the kind of pipeline that will allow us to mine scientific literature, not only one single type of entity, but different kind of entities that we think would be important to capture and their relationships. And so over the years, we have developed different tools, and we start developing some kind of common workflow where we can take modules and put things together to detect new data types that we are interested from the scientific literature. So we need to identify basic entities like genes, proteins, but not just individual gene, individual protein. We wanted to know about gene mutations, copy number variations, all those kind of anomalies regarding genes. Protein modifications, not just proteins, but if protein is phosphorylated, what are the kind of information can we extract from there? And we wanted to know how these entities relate to each other. Gene connect to drug, drug connect to disease, and gene connect to gene in the interaction network and so forth. And what's the biological context? Are we talking about this is a cell, data you collect when you do, basically you do experiment using certain cell types, or is this a patient population study and so forth? So we try to capture those kind of contexts. And then, because when you read the scientific literature, everything is written in free text. But if you wanted to integrate that information with information in databases, you need to find a way to know that this gene, this protein linked to that database entry. And so we have to do this kind of normalization. We need to do mapping of the free text in the scientific literature with IDs, numbers, unique identifiers in the databases. And so we do different kind of normalization. And so we have a basic workflow, but we are certainly most interested in what the end results of that information extraction will lead to. It will lead to us the capability to do more integrated analysis and discovery, to ask some core questions. And so we connect that in through the ontology in so-called semantic web environment to do representation in knowledge network, something that you can visualize, a biologist, investigator can visualize and can ask questions. And so we put all these different tools that we develop with our collaborators onto a central portal. And this is a poster with many tools that we develop. So certainly I'm not going to go over that, but just to give you a flavor of the kind of entities, the kind of relations that we extract from the literature. So we have ALINS P, TM for post-translation modification, but ALINS P allows us to extract from the scientific literature information about kinase, about substrate, the phosphorylation site. Ifib is a tool that built on ALINS P that allows us to say, okay, this protein is phosphorylated. It changed subcell location. It changes interaction partner. So what is the new interaction partner due to this phosphorylation? So that's what it does. Dymax, this is a situation where a tool that allows us to look into okay, how does mutation, how does this particular variant affect its disease state? And so that's a mutation disease association. MicroRNA target gene information. So we know that genes get regulated at the transcriptional level, at post-transcriptional level. MicroRNA has become a very important type of molecular entity that people have identified its crucial role in controlling the gene expression at the post-transcriptional level, and microRNA themselves could serve as markers, as disease markers as well. And so we were also interested in looking how microRNA play a role in this kind of gene-protein network. And microRNA in association with disease. Yes, it's associated with disease. Many breast cancer, they are identifications of microRNA that are associated with it. But in addition to find association, we wanted to identify the sentences and relations that indicate how do they actually associate. So we not only saying A associated with B, but how A associated with B. And that's the kind of information that we extract from the literature. And so we can develop every single special custom tailored tool for different purposes, but that's not good enough. We need to be able to, when we identify there are important molecular entity types that we wanted to look into, can we generalize our tool to do it? And so we have this generalization tool. Basically use, again, analysis of language patterns to syntactic variations, how authors write their articles, and basically use computational linguistic approaches, natural language approaches that allow us to automatically construct patterns for us to detect new entities and their relations. And so this is just an example of that rule-based, pattern-based system that allow us to take that kind of free text sentence and convert them into some kind of structural format, kinase substrate site. And so basically from free text to structure information that can then be represented more systematically. So this is to identify protein-protein interactions of phosphorylation. And so just using these two tools alone, just as an illustration, we can then construct knowledge network. In this case, RNSP kinase substrate site information, IFIP, phosphorylation-dependent PPI. So we can then draw a picture like this, where bad protein is a protein that is really bad, that has some disease consequences. And we can look at the different phosphorylated forms of this protein and what are the kinases involved in phosphorylating different sites of this protein. And once this protein is phosphorylated at different sites, what kind of interaction partners it may have. Because different interaction partners would allow this protein then go to, basically get involved in different signaling pathways and so forth. And so we can start collecting those kind of information. And so by doing this kind of PTM-focused text mining, we can extract a lot of information from scientific literature. There are also many curated resources that specialize in PTM. We bring them all together. We then create a portal where user will be able to look at the network of the kinase, the substrate, the interactions, and we can look at the conservation. We can look at is this variant, if this modification actually occur in mouse or in rat, if they are other variant in mouse that has some pathogenicity, how much can we translate that information? Is the particular site conserved? So we can look at all those kind of information. So we can take information from model organisms to understand human biology and disease as well. And this is just an example of the kind of things that we can do once we build that, the IPTM net using that kind of resource. And so for this is just one slide to show how we can use that resource for us to interpret beta-catenin, how beta-catenin could serve as a cancer driver. And so we take the cancer mutation data from Cosmic. So Cosmic has this mutation data on various types of cancers and from patient population. And so we know that people can cluster cancer based on ER positive, ER negative, expression of certain genes. But what we show here is that you can cluster cancer based on the modification site as well. So these are the modification, post-translation modification site. They coincide with basically the genomic variant site. And we can then try to look at if there's a modification, what potentially functional consequence of that modification may cause. And then can we use this kind of information to then try to interpret the clusters based on those sites. And so the key here is that the researchers have been clustering different kind of diseases at the gene expression level. Now we can cluster diseases at the individual site level, individual variant, individual modification, and so forth. And so that really is a key message, take-home message for this one. And so I just show you the phosphorylation information, but we have many other tools that allow us to look into much more disease-driving, causing associated protein entities and the different kind of relationship that we can capture. And so what I'm going to show here is that we can then, with that kind of knowledge network, we can then try to overlay the knowledge network with omics data. So this is the case where we have the data coming from the cancer genomics. So TCGA, that's Cancer Genomic Atlas data. So it has the genomic information, copy number variations, the variants and so forth, SNPs and so forth. The transcriptomic data regarding expression at messenger RNA and microRNA level. There are also clinical data regarding tumor stage, some patient demographic information. And along with that, also proteomic data, look at protein expression and the phosphoproteomics, how the phosphorylation information looks like. And so we just use as illustration one particular gene as an example, PDHA1. And this is an important gene. So the PDHA1, it is actually it's a phosphorylated protein phosphorylated by a kinase, group of kinase, family of kinase, PDKs. And there's actually a kinase inhibitor, DCA, that inhibit PDK so that through the inhibition of the kinase, therefore the inhibition of the phosphorylation of PDHA1, it could reverse the so-called Warburg effect in cancer. And so this has been tested in many cancer lines, clinical trials, and it has some positive outcome for certain cancer, but not for breast cancer. And so we took all the breast cancer data from TCGA and CPTAC and overlay those omics data from those patient population onto the knowledge network that we built. And this is just, sometimes it's difficult to look at this kind of network because it looks like a hairball, as people would say. But anyhow, this is PDHA1, the one in the middle, and it has different modification forms. Different sites would get phosphorylated or acetylated and so forth. And there are different kinases, PDK 1, 2, 3, 4, involved in phosphorylation of different sites. There are phosphatases that reverse the phosphorylation, and there are transcription factors that control the kinases. There are microRNAs that control the kinases. And so basically, it's a rather complex network, but bring together the different players that relates to PDHA1. And so I just wanted to zoom in to this one. So let's just focus on PDK1 again. So basically, it has many modifications, many enzymes control its regulation, but we can tease out for this particular PDK1, what are the kinases working on it, what are the transcription factors, what are the microRNA that actually control the expression, the activity of PDK1. And so you can look at the downstream event and upstream regulatory events all through this kind of network connection. And we can then overlay, once we have this kind of a priori network, knowledge network, we can overlay the kind of expression data from TCGA and CPTAC and see how we can explain the upregulation, downregulation of the substrates at the regulators level. And because the a priori knowledge is always incomplete, we can also use experimental data from all those large-scale gene expression, protein expression profiling experiments to identify new players that are important in this network, how they are correlated with each other, with different players. And so we can put this kind of quantitative information on top of the knowledge network to create new knowledge and to also try to understand the dynamics, the interplay between the different players in this kind of molecular network. And so we took that project, the text mining project, further for this Maze2K project. So this is a project where we are funded by the NIH, the so-called BD2K initiative, Big Data to Knowledge initiative, where we extend our text mining tool to look into genomic anomalies that relate to more in the clinical trial setting, in the therapeutic setting. So we wanted to know the predictive effects of genomic anomalies on therapeutic outcomes for, say, FDA-approved targeted therapy. And so we then extract all those important entities and concepts from the scientific literature. So we need to know, in this case, for drug, what's the intended drug target, what's the intended disease for that drug, and what is the patient population information, and what's the patient outcome, are there adverse events, and so forth. And so we extract all those kind of information, and we can tease out the relationship among those important entity types as well. And so this is ongoing, and certainly is quite exciting work. So in the last part here, I just wanted to show how, again, we can bring all this information to do some kinase drug inhibitor analytics. So again, we have our phosphorylation network. In this case, we use the Omics data from another major NIH initiative. So this is an initiative called LINKS, where they are collecting a lot of molecular profiles that relates to drugs, pathogens in the cell environment. And we wanted to see how can we, again, overlay this kind of publicly available high-throughput global scale data for us to make some interpretation in collaboration with our colleagues to interpret their trial data and so forth. And so LINKS, so this is, again, a major NIH initiative, multi-center, tens of millions of funds put into basically try to collect molecular signatures in terms of how cells respond to pathogens. And so in this case, the pathogens could be drugs, could be chemicals, could be genetic perturbation. And so a lot of data available, again, publicly available, more than 1,000 cell lines, 40-some thousand small molecules, and so forth. And a lot of data collected, again, global data, gene expression data, the proteomic data. They also have, because we were interested in looking at the kinase drug inhibitor analytics, so we were particularly interested in looking at the kinase inhibition information. And so, for example, there's this kinome scan data where they look at several hundreds of the drug inhibitors and how they individually inhibit the whole hundreds of kinases at the, in this kind of assay environment. And so again, a lot of global data available. We put all this information into a knowledge network. Very difficult to tease out, but I just wanted to take you to look at, zoom in to one particular aspect. So for this network, this is a sub-network of the previous giant hairball. So it is a knowledge network decorated with information on Dinosaur Club. So again, it's a kinase inhibitor, and so the triangles, these are kinases. The round circles, these are proteins get phosphorylated, and the green ones, meaning that the phosphorylation level is reduced. The red center, meaning that the phosphorylation level is not inhibited. The green and red outside, that's the gene expression level. And for kinases, because all the triangles are kinases, if a kinase has a big triangle, meaning that their activity is inhibited in this kind of scan data. And so what we find is that most of these big triangles, they connect with green circles, okay? Meaning that most strongly inhibited kinases, they are associated with downregulated sites. So when you inhibit kinase, if you look at their phosphoproteomic data, the phosphorylation is also inhibited. However, we do see situations where that's not the case, where we have red, the one here, this is a red center, but this is a big triangle. And so if you look at it, it actually is also connected by this small triangle, meaning it's a kinase not inhibited. They are still, this kinase is still active. And so this kind of information allows you to look into the crosstalk as well. So you can view this kind of data, visually. A biology can, quickly look at this and get some sense out of it. But we can do global analysis and directly output the kind of information that is much more, summary information. So you can chart the entire thing to look at how the kinase correlate with the kinase inhibition correlate with the phosphorylation level of the substrates. And you can then start teasing out, in this case, is Dinosaur Club. What are the kinases that potentially is active in that kind of kinase inhibitor environment, what are not, and what are the technical reasons or biological reasons that certain kinases may not perform as what we would expect. And oftentimes, it's those unexpected results that really leads to interesting finding. So we continue to work on this, and we continue to basically develop a global system where we can look at hundreds of drugs and the different cell lines and the kinases in a very systematic way. And so this takes us to this final collaborative work that we do with Dr. Wu Guha at NCI, National Cancer Institute, where he's been studying lung cancer, non-small cell lung cancer, where he has worked on previously study on first-generation target therapy and recently on third-generation target therapy. And so we all know kinase inhibitor is notorious in that it initially works, but then resistance develops, and oftentimes the resistance developed is due to additional variants, additional mutation that occur on the targeted gene and so forth. And so this is an example where this particular drug, Phosphorylation-dependent, which the clinical trial was stopped because it was the resistance to that particular target variant was developed. And so we worked with Wu in looking into how can we explain the resistant versus sensitive cells in terms of the kinase, their substrate activities. And so we do all this mapping exercise, and we were able to identify kinases that may affect certain sites get upregulated, and kinases for sites that can downregulated. And the nice thing about this is that because you do this kind of global analysis, now you can start thinking about, is there a way for us to manipulate this so that a resistant cell would look now more like a sensitive cell? And so that would allow us to try to identify the kind of target that would be candidate for combination therapy, in that the resistant line could be converted using the opposite patterns of the kinase phosphorylation basically expression. So I kind of run through my 40-some slides quickly. So acknowledgement to a tremendous team that we have and all our collaborators, and certainly very generous funding support from NIH on many different aspects of what we work on. Our colleagues in the computer science department, CBCB, PI team at University of Delaware, and also I still have some faculty member at Georgetown University. We continue to work very closely together. Many of students in our computer science program, in our PhD program for bioinformatics and systems biology. And finally, if you are interested in learning more about the kind of things I'm talking about, we just launched this Applied Bioinformatics fully online graduate certificate program, 4 courses. Go to our website. So just if you think this is useful, you can consider or spread the word. Okay, so that's all I have. I would like to entertain some questions here.
Truly amazing and most informative, not to mention overwhelming. So historically, most pharmaceuticals have been found by serendipity, pure luck. But that's really no longer the case. It's now a matter of really deeply understanding the biology. But the biologic system you portray is awfully complex.
It's complex,
so
and
we're in thinking about therapeutic targets. Where do we begin? How do people go about figuring out what's the best thing to do?
I think the nice thing about informatics approach is that even data is overwhelming, you can really dissect it, vertically, horizontally, any which way that you think makes sense. And then you can really zoom in. The nice thing is that, like the Google map, Earth map, you can zoom out and zoom in. And you can really zoom in and see, important subtleties in subnetworks. And once we build this kind of global network using reference data, we can start overlaying that with individual patient data. And that's where, or we can look at the subpopulation of say, certain patients do not respond to certain drugs, and we can bring the data and try to overlay onto this and try to start teasing out what could potentially be the underlying molecular mechanism that leads to those kind of outcomes. Yeah,
this is a very wonderful talk. I would like to know, how can you combine the bioinformatics techniques and methods with clinical data for the outcomes research and comparative effectiveness?
Yeah, excellent. And that's something actually, a proposal we are writing at this time with Andy Cope on sickle cell disease. And so there's this wonderful NIH initiative. It's called TopNet. I don't know, well, you probably heard about it. So TopNet is an NHLBI initiative where they generate a lot of data on 100,000 patients, a cohort of 100,000 patients, and they also collect the associated Omics data, along with clinical data, electronic health record, and those kind of information. And the idea is that again, to connect, I didn't really go too much into that other than the drug work here, in connecting the genotype with the phenotype. And so we were, working with Andy, looking at sickle cell disease and all these complications, the comorbid conditions. And so here on one side, we have all this the network of the phenotypic relationship. We can see sickle cell disease, it has all these phenotypes, and what's the dependency among those phenotypes. And then here we have the genes, and we know through GWAS, through other type of genetic profiling, certain variants, certain genes are connected with certain phenotype. And so we can start building a network, connect this together. And so, yes, when we wanted to get much more fine-grained and clean data regarding phenotype, that's where a lot of those clinical information is important. And so for the phenotypic computing, use Tim Bono's word,